So today we're going to talk about iNaturalist. We're going to talk a bit about what is citizen science, which iNaturalist is a citizen science project, and a little bit of a history on citizen science. Then we'll have a look at iNaturalist, and after that we will go through how to install it and how to use it and get the most of it so that you're encouraged to keep going with it. So background. Citizen science is a word that's been around for a little while. What does it mean? Well, what is it? It's, in the simplest term, the ordinary person recording their observations. The byproduct of this, it becomes data for scientific investigations as well as provides data for decision making. So, a little bit of brief. To start off with, there are a variety of terms for the word science, but citizen science is generally accepted across the world. It's a modern word, a modern iteration since the mid 20th century. But let's have a look at a bit more history. So, many people would have heard of the Christmas bird count, began in 1900 and has been going continuous since then. We need to remember the word scientist was coined in 1834. So, that word hasn't been around for very much longer than the word citizen science. Let's keep moving back through history. So we now go back to Robert Marshman. He started recording 27 indications of spring in the United Kingdom. When did he begin this? 1736. His family kept records until 1958 when the last of the Marshmans died, but the legacy of what they left behind has continued. Marsham is known as the father of phenology. And this is really what a lot of we're looking at with iNaturalist. But we can go back even further in history. Most of us have heard of the Royal Society. This was begun in 1660. We tend to think of the great scientists of the day, Newton, Kepler, etc. I mean, one of the members was Thomas Povey, and he was a merchant politician. So he was a citizen scientist in the work that he did. But we can go to another one, the Kaito cherry blossom records. When did they begin? Soon after Kaito was actually founded as a capital of Japan back in about the 8th century. So records were a bit patchy for a while, but since the 14th and 15th century, there's been continuous records. It really is amazing how long we have, as a people, been keeping records around the world. So science really began with citizen science. So let's move on to iNaturalist. What actually is iNaturalist? It is a citizen science project, as I said before. It actually makes use of the principle of crowdsourcing of observations. It has objective, verifiable data, which is very important for science, and it's not dependent upon the observer's knowledge base, just their curiosity and desire to want to record and know what they've actually seen. So, iNaturalist is a collaboration between the California Academy of Science and the National Geographic Science Society, and it began in 2008. So, how big is Naturalist? This is a map of observations taken up until 14th of April 2023. So, it's worldwide, truly an international project. How many observations? As you can see there on your screen, 132 million observations, 2.5 million observers. They've found 423 species. So this is a really big project. What's interesting to note is that not everything does get identified to species level. About 74% roughly, or 3/4 of the observations are there. And this is because iNaturalist is picking up observations of life that we haven't documented before, and they're waiting to be described. So another aspect of that's worth to think about with iNaturalist is yes, there are a lot of users, but if you see the red line there, not that many are really active. And the aim of this talk is to encourage people that they can be active members on this site. So, iNaturalist basically works upon the principle you record your observations primarily on a smartphone. You upload them and you share them with others on the site. And then there's a bit of a discussion and a determination of an identification. But then what happens with all of this that's sitting there? Well, they go into the biological databases, both nationally and internationally. The national one is the Global Biodiversity Information Facility, which is a joint project of nations around the world. They all contribute to the maintenance of this facility. And for Australia, we have the Atlas of Living Australia. So your information doesn't go away, it is kept and used. So let's talk about installing the app. iNaturalist can be installed on your PC, and it's just a simple matter of following the instructions in the sign up there, or you can use put it on your phone, in which case you've either got the Google Play or App Store, and in effect you follow the instructions as given there. If you do run into any problems with installation, you can actually contact iNaturalist via an email, which will be on the last slide. So let's talk about iNaturalist on the phone. I'm primarily going to use I have a Samsung, so I will use that one for demonstration. So there will be some variations, but the general principles are the same. You'll have an icon up in the top left-hand corner. If you click on that, you get a drop-down menu of all the things that you can do on it. We're going to actually walk through how to do an observation now. So there's a little green button, which you click on this button and it'll open up a window and it'll ask you, what type of observation do you want to take? And you'll notice you have a choice. You can go without a photo, but this will always remain as a casual observation. Sometimes that is useful when you just want to have a record of where you actually were and you may have taken the actual image on your camera, which you will upload later. You can take a photo directly from the camera, from the phone, or you can choose an image that's on your phone. The other type of observation choices there, of course, are sound. Once you've selected that and it's uploaded, it'll ask if you want to take another image, and you can add as many images that you want to help with your your observation. Next thing is, what did you see? Now this is a very interesting little icon. You click on that, and you'll get a drop-down menu of what the computer vision thinks you've seen. I'll talk a little bit more about that further on, but just for a simple thing here, you'll notice it says we are pretty sure it's in this family. It's also given some suggestions for some species, but I wasn't confident in my identification, so I stuck with the family level because I knew it was a gecko. Then you'll have another tab to be aware of is your location. Now, sometimes the location with phones today, locations are really, really good, but you will get occasions when that's not good. So it pays to your location, make sure it's in the right place. Where you may have problems is if your phone's been off and you take the image too quickly and it hasn't had time to get the coordinates for where you actually are, or there's been very thick cover such as your canopy, or it might be heavy cloud cover, and that could throw out where it is. But it's a simple matter of just moving the crosshairs icon to where you know that you are. You can also do that on the computer later. You can do some editing, which I will talk about later. Another icon to be aware of here is your location visibility. By default, iNaturalist sets everything to open. Now you may not want that. Your other options are obscure or private. If you click on private, then no one can see where you have been at all. But that sometimes is not helpful because identification of some species can be dependent upon location. If you click on obscured for the Adelaide area here, that will obscure 1 degree, which is the equivalent of about 20 square kilometres. The other thing that will happen is that if what you have photographed does have a listing, a legal listing, such as threatened or vulnerable, etc., iNaturalist will automatically obscure those for you. So having done that, you just click on your tick button there to save it, and it's done. So let's have a look at the website. On the website, once you've signed up, you open up to this lovely page, which they will constantly show different observations. Some beautiful pictures turn up here. So of course you just log in. You can also use either Facebook or Apple or Google as alternative methods of logging in. And the first page that's going to open up is what's called the dashboard. Now this page has quite a bit on it. Across the top are all the various tabs. Now I'm not going to go into those tabs just yet, but I will point out this widget down here at the bottom. It has your getting started guide. So if you ever have if you want to go over how to do things, or you're not sure how to do something, you click on the link in there. It will take you to a whole new page with lots of topics, frequently asked questions, videos, tutorials. Very, very helpful. You can get to your dashboard page by clicking on your icon, drop down menu, and just click on dashboard. So let's have a look at where all our observations are going to be sitting. So it actually says your observations or my observations. And this page has a lot of things on it. So I'll just point out a couple of them. First up's your icon, as I said before, with the drop-down menu. There's the speech bubble. This is the one that's you'll probably use quite frequently because here you are any messages, any communications where people have looked at observations or you're in a conversation about a particular observation, you will get notifications there. The next icon, the envelope, that's generally is private communications. If you want to talk with a curator or another iNat member somewhere, you can send messages and have conversations. So that might be things that are not actually relevant to a specific observation. Someone sent me a message where they were wanting to get access to some information that I had, which had nothing to do with any of the specific observations, and that's a good form of communication. Another button to be aware of is this green upload button. I will come back to that later with a whole section of what happens there, but this is when you've got observations that are on your camera or historical observations that you want to upload. So just coming down here, you'll see two different icons. One is a drop pin and the other is a dotted I. The drop pin means that that location is open, it's visible to anyone. The dotted I is that I have obscured that, and that is only available to be seen by those that I give permission to. And I also, of course, will see that. Over on this side, we have our species name, and the green bar says it's reached research grade. So research grade is when there's a 2/3 majority, and I'll come back to that again a bit further on in one of the other pages. So searches. One of the really good things about iNats is you can search For your observations, you can search for a species, all sorts of different things. You'll find the search bar up here. This will get you, say for instance, you have a particular species in mind, like you want to know where all the Thelomytra alcockii are to find out their flowering times, etc. I'd type that in. That'll open up an observation page with just all the Thelomytra alcockii, for instance. Or the other search bar down here is when you're looking at your own page, or when you're exploring and you want to search within a specific page, that will also open up. You type in your species and that will take you to that. And of course, you can do a search for location once you've got, whether you want to do just locations of your observations, a specific location of all your observations in that area. In this instance, my observations are sitting in all my observations around the world, which isn't very far. Another thing in search is filters. And in the filters, as you can see, you can select what type of search you want to do. You can search by dates, exact dates, range of dates. You can search by months, so what's around in a particular month of the year. You can search by categories. The dotted one there is for the unknowns. You can search by the quality of the observation, whether it's a research grade or needs identification. So you have quite a choice. So let's look at a single observation page. What information is on there? As you can see, there is a lot of information. So we'll go through this page in a little bit more detail, starting with the top of it. Now I'd like to point out here at the very top in the URL bar, the address bar, every single observation has a specific number, and a unique number. And this means you can use this when, say, for instance, you're having a discussion and you're comparing a couple of different observations. You can direct people specifically, copy and paste that in, link into your conversation and direct people to a specific observation. There's also other reasons. I'll give another example of that further down. Here will be your first suggestion of what you think your species is. I will come back to that. Then below that comes a whole conversation. And in this instance, we see that there was a disagreement. And so it's a bit of a conversation. And I actually asked this person, what do I need to photograph? What information needs to be in those images? And they were kind enough to tell me what I needed to photograph to help with identification. So that is a really good thing when you're helping people. If you're disagreeing with an observation, an identification, it's helpful to say why, what are the features you're looking at, and this is how we all learn and help each other learn. Further down is all those green ticks. As those green ticks are passed, when all of them are green, no red crosses, it goes to research grade. In this instance, for this particular species, we actually only got to genus level because I hadn't photographed the distinguishing features. And I asked the question, can we get this to species level? And the answer was no. So you can click on, no, it's as good as it can be. And I did that, and that actually did go to research level. Another feature on this page, there goes your 2/3 majority. So the way this works in the simplest form is that if two people agree, so say if I put up the correct identification, someone comes along and agrees with me, that's 2/3 majority, and that will go to that. But someone else might come along and say, no, I think both of you are wrong. And they will disagree and put up what they think it is. And the process starts again. And then you have the conversation, other people will join in. And when 2/3 of the people in discussion come to an agreement, it will go to research grade. Uh, that's it in the simplest form. If you click on the About button that's just below that green line, that gives a much more detailed explanation of how it works. It's quite a robust system, and I've been very impressed with it. So there's my About button that you need to click on. Like I say, that will bring a drop down explanation. Now, this next one is some annotations. This is done for plants and animals. They have slightly different annotations, but it's also very helpful to be able to state whether your organism is alive and where it sits in the scheme of things. A lot of people don't always fill that out, but it is a helpful thing. It's helpful with searches, and I'll show you that on another page. Also, projects. You can join projects. I won't be going into projects towards the end of the talk. I will briefly mention two key projects that occur annually here in Australia, or particularly South Australia. We'll get to that later. And the other thing that's worth knowing about is top identifiers of whatever. Now, this is very helpful when you're having discussions and you have some uncertainty. You can actually contact one of these people by tagging them at whoever, like in this instance, at Kevin Bontham, and they will get a notification. I do find that when I am tagging people, I like to have a look at their profile and just check where they're located, what their level of expertise is. Over time, you get to know different ones and who to tag. Who's the appropriate person. So if I'm looking at an orchid, for instance, in the eastern states, I know that there's a lot more similar species. And so whereas here in South Australia, I might clearly identify something as Pterostylis pedunculata, interstate, there's Pterostylis erecta. So I'm not sure of my difference between the two. I would actually contact someone that I can see from their profile that they know their orchids and can give the two, can differentiate between the two. So it's a really helpful thing to know about. So the species info page. This is a very good page for learning more about a species, and is a lot of information on here, and we'll just walk through it. So to get species identification page, you need to click on the genus name, the species name, whether it be a genus or species. That will take you, open up the page that will look either like one of these two pages. And this is on my settings, it is at the About tab. And you'll notice that one of the tabs here has a lot of information on it. The other has what looks like a code. It's a template. And this is because this needs to be filled in by an appropriate person as the observations are added to it. And iNaturalist has a template which people can follow. It's really good because it generally has to be referenced. You can check the sources of the information. Primarily, they use Wikipedia, but it is referenced. So it's more or less like having a little book there with you. Another thing is this two sections down here. So this is in the computer vision. And as I mentioned before, the computer vision will suggest potential identifications, but it will not suggest something if it doesn't have enough observations. So the grey button says pending. The Atratrarchy prostrata has only about 60 plus observations. That is not enough for the computer vision to use that confidently. whereas the dusky miller on the other page has a green saying that the computer can recognize it. And that is why sometimes when you do an observation here in Australia, because we don't have enough data in there yet, it will not bring up what the species is. And it's something, the more we populate the data, the more confident the computer vision is at identifying what we have photographed. Another tab. That's what I didn't do. So just looking up here, that tells you, yeah, there's only 63. So let's look at some other tabs. You've got one on a map. You can see where things are. Now, maps are very handy if you're not sure about what your observation is and you think, yes, this is something I did. I thought, yeah, I photographed a wren. The computer vision suggested wren. I saw the word and I clicked on it. But I should have actually checked the map because I didn't realize that wrens are a northern hemisphere family. We have fairy wrens. So maps are helpful just to double check, are you putting down the right identification? If you see that the name that's suggested and you think it is that and you see that it's in nowhere near where you are, chances are it is not that species. So, and yeah, you get an idea of distribution of things. Another icon here is the taxonomy. And you can click on any of the levels in the taxonomy and see where your species sits. Another icon is similar species. So this similar species is not based upon what has been written in descriptions so much as it's based upon other iNats observations that have been misidentified as a particular species. I find this is useful to look at because if I find there's a lot of things where it could be confused with, then I tend not to make an identification at species level. So another tab, this tab tends to be open by default and it's a seasonality. So you can see when you're likely to see a particular species. But you can also go on history and you can see how many observations have been taken, whether they've reached research grade and when they were taken. So as you can see, even though there are a lot of observations, most of them are more recent. And our final tab here is, in this instance, the plant phenology. And you've got the flowering time, etc., etc., and that's helpful. You can do searches for whatever aspect that you are wanting. So now we move on to the profile page. This page is, I did mention before when we're looking at top identifiers of X, is where I go and have a look at who is the person. So here's my profile page. It has I've put up a picture for help people identify me. It has my observations. It has how many species I have and also how many identifications I have done of others. Probably the most important thing about your profile page is the information about yourself you put in here. This helps others to know whether you're able to help them with answering a particular identification. It's obvious from my page that my interest is Australian orchids. Over time, I will probably add more information on there. And some people do, they update their profile every year just to say where they're up to. It's a very helpful page to have. So enough of profile, let's move and we're going to look up uploading. So not everyone takes pictures with their phone. They use their camera. Or they've got old pictures, and you can, if you know where you've been, upload your historical data. So you click on that green icon that I pointed out to you, and you will get a page open up looking something like this, and it says drag and drop your photos or sounds over. Quite simple. We do it on quite a few different things. Then here we are. I've dropped in a couple of 4 images, and over here it says submit 4 observations. If I click on the submit button now, that's what will go in. But there's a little bit of a problem here because these 2 are images of the same plant. And they should go in as 1 observation, not 2. That brings it back to 3 observations. But this one has 2 species in there. So I really do need to split that up, and that will bring it back to 4 observations. Before I do that though, I can put in location. So if I click on the location field, up pops a map. You zoom in your map and to where you were, and you put in there'll be the circle, and you can make that circle large or small as you like. So this one, it's given I've made it to the size of a 79, almost 80-meter radius because I was somewhere in that area. I can't be more precise than that because this is an old observation. You click on update, and that's location done. So now all of them have their locations, but we've got to deal with these 2 images. So if we click on those 2 images, then up the top here we see combine. You click on combine, and they make that now 1 observation, and you'll see it says on there there's 1 out of 2 showing on there, and so now we're down to 3 observations. But we've still got to deal with the end one, which has 2 species in it. So you again select that, and then you click on duplicate, and that will create 2 images. You can fill in on the fields there what the species are, but also make a note in the notes what it is you want identified. So in one you say you want the orchid identified, and the other you can say that you want the sundew identified. That is really helpful for people coming along and helping. And of course now we do need to fill in the species for each of them individually. And we are now ready to submit 4 observations. That is 4 observations with 5 images, of which 2 belong to 1 observation, and another one is being split up into 2 to get the 2 species in. So they were photo observations we've mainly looked at, but also there are other types of observations we can get. I'm just going to briefly mention them. They can be of photographs. We don't have to necessarily photograph the organism. It can be an audio, it can be bones, it can be tracks, scats, feathers, nests, diggings. As you can see, quite a lot. In fact, I haven't put everything down here. It can also be diseased creatures or evidence of infection. So in this instance, some people who, when they got COVID, they photographed their rat, and that was accepted. Um, if you've got an animal, for instance, a budgie, and it's got a disease, and you can see evidence of the disease in the bird, you can photograph it and ask for the disease to be the observation. So, but I didn't mention videos in that list, and there's a reason. Videos are not supported in iNaturalist. You cannot put a video observation up inside iNaturalist, but there is a way of dealing with videos. That is, you take your video and upload it to your YouTube channel. I have a playlist on my YouTube channel that I call iNaturalist Observations. That's where I put them all. But I also take a photograph and I make an iNaturalist observation as normal. Then I copy and paste the link to the video in the notes section. And I also do the same from the iNaturalist observation using that unique URL and put that in my in the description in my video. So there you go. I've written that in the notes section. There's the link, ready to go. There it is, the iNaturalist link in the YouTube video. That's how you can get an observation up. Now that can be very helpful. Sometimes the behavioral pattern, particularly with birds, it might be a particular way a bird flies. It could be insects, the way they're behaving. It just adds extra information. And the thing about this with your image that you put up, it doesn't actually have to be clear in this instance, particularly if your videos are very clear. So let's have a brief chat about editing. You can edit in either one of two ways. You can edit on your phone. There's the little pencil icon up there. You just need to, in your observation page, click on that. So you can edit various aspects of your observation, but in this instance, I've gone to the photo because I want to crop the photo or just make a few changes. And again, you go to your photo and you see your editing icon, and then you have a page that opens up and a lot of different things you can select. So you can crop your image, you can increase your contrast or your sharpness or brightness. So just to make it that little bit easier um, for others looking at your image. You can also edit on the website. I won't go into a lot of it, but the page is basically on your observation page. You click the edit button and then you'll have something like this opens up and you can just go into whichever one of the fields you are needing and edit it. And then don't forget to save your observations. So that's editing the website. So now I just want to give a bit of a mishmash of some tips and hints on doing observations and identifying observations. So when you're out, stop and just stay still for a while. Specifically, I'm referring to plants, but it can also be just keep still, observe for a few minutes. On plants, there might be insects present, or there might be some galls or things that are not so obvious, some unusual growths. With animals, by keeping still, it gives a chance for you to observe them without them being scared off. Look for unusual growths on plants or animals. So, as I said before, it could be galls on plants, on creatures like with sleepy lizards, there's ticks. That can be a separate observation. Check the leaf litter. Most of our species are down low. They're going to be down below knee height. So don't forget to look low and also look high. Look up in the trees, look down in the bushes. You'll be surprised at what you can find. The other is when you are doing your observations, take multiple images of different features from different angles. I would err on taking a lot more images on something I don't know um, in case I would, on the chance that I do get the identifying features. Do things close up, but also do distant images. So particularly with trees, get the whole tree in from a distance if you can, but also get close up to the tree. Get the bark, the way the leaves grow, the leaf itself, the fruit, the flowers, whatever you can. And the other is indicate with some type of scale, your hand or a car key. So we're not about getting beautiful pictures, we're about getting pictures that give us information. And putting your hand behind a leaf gives an idea of the size. And if necessary, crop or edit the image, as I showed you before. Audios. Audios are interesting. You do need to get a reasonable amount of time, especially for the frogs. Birds, you may actually have to go a bit longer. Sometimes with bird audios, you don't have just one species there, you may have several. And I might do, in some instances, a 2-minute audio and then say at 30 seconds or at 68 seconds, I would like it identified at this point. Yes, specify the organism you want identified. So if you've got a whole lot of different things in a picture, but you're really only concentrating on the plant, say you want the plant identified, or if it's a beetle, say that that's what you want identified. The other is ID to the level that you're comfortable with. It doesn't have to be species level. People will come along and help you with the identification. So it's okay to just put down plant even or insect. I do that with quite a few of my observations. Someone will start to add and will gradually bring it down the taxonomy level. Another thing to be aware of is if you've put up an observation, you didn't know what it was, someone comes along and says what it is, don't confirm that if you're not sure of your ID skills. Just because one person has said it, they may or may not be right. Leave it for others to come along. If you're happy enough and you are sure, you've gone in and checked, say, that species page and you think, yeah, that doesn't look like anything else, it has to be that, that's fine. But you don't have to confirm if you don't think you could identify that by yourself without someone else first telling you what it is. So they're just some hints, tips and hints to keep in mind. There's probably a lot more, but that's some to hopefully get you going. So I want to come back to projects. In projects, there are two types of projects in iNaturalist, what we call a traditional project and an umbrella project. I'm not going to go into all of that. That's a whole big subject of its own. But a traditional project is one that you need to join. They will often have extra things within the project, for instance, fungi map. They will have questions. So it's not sufficient just to take a picture of a fungi, but they've got some questions to be answered. I think there's about 3 questions there. The other type of projects are umbrella projects, and they work on the basis that your observation, you do not necessarily need to join, but your observation will be gathered into that project if it occurs at the time of the project. 2 projects that I'd like to mention. One is the Great Southern Bioblitz. This is held in countries around in the Southern Hemisphere in spring. Each year it's a slightly different time. But if you go to Ferox Australis, you can get information on it. And within iNats itself, you can get information on when it's going to be held. So for this year, in 2023, it will be held towards the end of October, beginning of November. Another project that happens, this is an international one. It's held in the Northern Hemisphere spring, which makes it our autumn. So this year it's from the 28th of April to the 1st of May. It's called the City Nature Challenge. Again, it's a worldwide project and it's a combination of getting some good data as well as having a bit of fun and seeing who gets out and gets the most observations. So there's leaderboards and this type of thing. The advantage of these 2 projects is that because they're covered on a weekend over a specific period of time, people tend to photograph everything. Rather than just photographing something that's unusual, they photograph everything to get the numbers up. And that gives us a far better picture of species in an area. And that's one of the things about taking observations. Photograph all and sundry because you never know what you'll photograph. I took a picture once of some white spots on uh, the leaves of a native apricot. Didn't really expect to get it identified, but there was a researcher on who was researching whiteflies. They identified it to species level because the cycle of the whitefly stays on the one plant. The interesting thing about it was that that was the first observation on iNaturalist. And when I checked the atlas, there had been 2 specimens collected and kept at the Northern Territory Museum, and they were collected in 1958. So there's not a lot of records. Are they common? I don't know. But that's the thing about iNats. You photograph whatever you see, you just don't know what you're going to be seeing, and it's just a great surprise sometimes. So, just finally, this final page is just a list of resources. You can stop the video and do a screenshot of it. Um, these links in here have a lot more information than what I've talked about, but I'll just point out there is, if you do have trouble logging into iNaturalist at any time, you can email them. The email address is there. For websites, Ferox Australis is a very good place to go for information on what's happening with iNaturalist in Australia. YouTube, there's iNaturalist has its own YouTube channel. The Great Southern Bioblitz has its own YouTube channel, so does Ferox Australis. So there's a lot of information on there. And just as an interest, there's a very interesting video of IA, the IA of iNaturalist as a tool for conservation impact. It's a talk given by a chap in America showing how iNaturalist has helped in some planning. They had a situation of a salamander that was threatened, and primarily threatened with roadkill. They were able to use the information from iNaturalist to determine where best to put their culverts so that the salamanders could go under the road instead of on top of the road. So that's just a little story on how helpful iNaturalist, the data you put up, can be. So thank you for taking the time to listen.